mosquitoes have been found in Iceland, and live mosquito, female and male both. Now you imagine what is going to happen. And only 3 or 4 mosquitoes have been identified. Now, first question is, how did those mosquitoes go there? So you can say, okay, flights are going through flights and other, and human is also. Now the problem is, once these mosquitoes start breeding there, all those which are vector-borne diseases through mosquito, those probably Iceland never heard of, now they will start. Now mountains, the temperature is increasing. Extreme heat is one of the worst effects of the climate change. Imagine in this room, after 1 hour, it is unbearable to sit, even with the AC. And in last 100 years, the temperature of the globe has increased 1 degree Celsius. Do you know what is the estimation for the next 100 years? It is 2 to 3 degrees Celsius. Imagine 1 degree has caused what? Now if it increases 2 to 3 degree, you can imagine what is going to happen. Glaciers are going to melt, all the low level, probably half of Bangladesh will be gone. So now this also has a excellent connection with the infectious disease, because now what is happening, because of the change in temperature, those places, some of the diseases, infectious diseases are not there because of the lack of the vectors. Now vectors are moving in those areas because the temperature is going up, and slowly this is going to cover. No matter what you do, unless we are careful about the environment and do the way we should treat, you don't have to do anything. By default, everything is going to collapse. Now India has, out of 100 countries, this also became a news last month, out of 100 hottest countries and cities in the world, India has 50. Imagine the impact what is happening. And that has a direct relation with the antimicrobial resistance, because there is a paper in Lancet which has shown that antimicrobial resistance 10 to 20%, they are connecting with the climate change. So now the temperature is increasing, AMR is increasing, with that climate change multiplying. So unless you have new antibiotic, and I can tell you the last antibiotic which was discovered, antibiotics were discovered mostly in 60s, 70s, last decade. After that, there are not many antibiotics have been discovered. So the existing antibiotics are getting resistant. If you don't have new antibiotics, it's really, really scary. I don't want to make you a very gloomy situation here, but that's where we are leading into. And particularly this north zone and the south pockets. And along with that, what is happening? Humidity. Once the humidity also increases, the infection also, so that's why they say, during winter, during rainy season, if you have a scar, it takes longer time to heal. The reason being the infections also, it is very difficult because of the humidity, because this pathogen also, in the dry, you won't see even a single mosquito when it is 43, 44. But as soon as the temperature drops, humidity comes, these are coming, along with that, the infection spreads. So therefore, it is important to understand the mechanism of the disease. And what is the mechanism of the disease? Like, I am only talked about the gastric ulcer, but this bacteria also causes cancer. Gastric cancer also, and adenocarcinoma is also because it prolonged stay there, and it also secretes many of the pathogenic factors which go to the cells which are adjoining, and then they transform the cells, which can lead to the genomic alteration, chromosomal alteration, and can lead to the cancer. Now therefore, what you need to do, that, as I said, there is a protein called KAGE protein which goes inside the cells and it transforms the cells, and that can lead to stomach cancer. And that's why WHO has recognized it the only bacteria which is a cancer causing agent. All others are chemicals and other things, but this bacteria can cause cancer. Now what do you do? Now this is the preamble I gave you. What is the importance? If you have infection, what you should do. But the question is, can I have something to treat this bacteria? Because what happens with antibiotics? Antibiotics are getting resistant. So therefore I have to constantly challenge myself and try to find out new and new target. If I can find a target which is present in the bacteria but not in the human body, that's a good target. Because if the target is present in both places, then it becomes very difficult. So this is a simple picture which was taken. E. coli chromosome, all of you know E. coli. E. coli chromosome is circular. And John Keynes, he saw this under electron microscope. And remarkable. You can see a circle, and within that one circle, isn't it? A circle inside the circle. So what is it? This is E. coli chromosome is replicating. So there is only one replication origin in E. coli. From where? From one chromosome it becomes 2, from 2 to 4, because that's what happens in all cells. When they multiply, the nuclear content has to be also multiplied. So therefore, this mechanism, as you can see here, this is a circle, and DNA has how many strands? 2 strands. Now if it has to be replicated or some protein has to be made by transcription, you have to unwind the 2 strand. Now imagine you are trying to unwind this 2 strand like this, so you have to put force. And how does that happen? There is a specialized enzyme called helicase which unwinds these 2 strands. Helicase is nothing but how the 2 strands are attached to each other. This is through base hydrogen bond. And if you want to break hydrogen bond, you need energy in the cell which gives you energy. ATP gives you energy. So this is an enzyme which is an ATPase. So it degrades ATP and the energy is come, and that energy is used to separate the 2 strands. But imagine you are trying to unwind the 2 strands, but they are like this. So after certain time, there will be positive supercoiling so much you won't be able to further unwind it. And that's where another enzyme comes which is called gyrase. And what does gyrase do? Gyrase is nothing but it comes, and when there is a supercoiling here, so much supercoiling tightened, the helicase cannot further go ahead. It comes and cut. Now if there is a cut, what will happen? The tension is released, and not only it cuts, it's also ligate. So gyrase in bacteria is the only enzyme which, it's the type 2 topoisomerase, which can relieve the positive supercoiling, and then it ligates it back, and also can introduce negative supercoiling. So then this is a very good target, and this gyrase is not present in human body. So if I can target this, then what will happen? I can kill the bacteria. So you have heard of ciprofloxacin, CIP, very commonly used antibiotic, isn't it? So what does CIP do? Nothing. CIP actually targets gyrase. How? It forms a trimeric complex. So normally gyrase cuts and religate, but CIP comes, ciprofloxacin, it goes and binds to gyrase. Gyrase cut the DNA, but it cannot religate. So in the presence of ciprofloxacin, slowly all the bacterial chromosomes will be cut, they cannot religate, so slowly they die. So this is an excellent drug, ciprofloxacin, but also excellent these days as an AMR because of rampant use of ciprofloxacin. Now many different versions are coming, but this is also causing lot of antimicrobial resistance because of this use. Now this is what a model of replication. Now little bit of biology. As you know, DNA has two strands. That strands has to be separated and replicated. So this is leading strand, continuous, and this is lagging strand. Why this is? Because you cannot replicate both the strands at a time. This because polymerase goes only from 5 to 3. Only this direction. So therefore this is continuous, but this strand will replicate it as and when it is getting unwound. So this is continuous replication and this is discontinuous and this is the lagging strand. So this is leading strand and lagging strand. Now you see you have one enzyme called helicase which is unwinding the DNA. That can be a good target. One enzyme is a gyrase which can be a good target because without gyrase after certain time the whole process won't work. And this is exactly we are trying to understand in this bacteria. So after giving this introduction, now I will discuss little bit what we are doing in the lab to understand the replication of this bacteria and why it is important, because if we get some new targets, we will be able to target this. So when we started working on this bacteria, we have shown you that circle within the circle, so that's our application origin, but there was no data where replication takes place in this bacteria. Not only that, some of the proteins like KAG, VAC, these are pathogenic proteins, they were studied properly, but the basic biology of the bacteria were not studied. Now what we found, interestingly, that a protein called DNA-C, and what is DNA-C? It helps the DNA-B to load on the DNA. And DNA-B is structurally hexameric protein, which is a closed circle. Now imagine this is a circle and this is the DNA circle. If it has to go there, can it go and load? No, because both are circle. They can be aligned this, but it cannot go and load like this. So what has to be done? First this circle has to be broken and then religated. And this is exactly this protein does, which is called DNA-C, which helps the DNA-B to come to the DNA and load on there, so that now this helicase can slowly unwind the DNA. But when we looked at the genome of H. pylori, we did not find DNA-C. So helicase is there, but the loader protein is not there. How is it possible? So that means either this protein can be loaded by itself, or there is some other protein which helps it to load. And we find that there is a protein called ORF 0897 which is not present anywhere but only present in H. pylori, and that helps this protein to be loaded. So that means since this target is not there anywhere, this is also a good target because it's not present in the host. And this is what we showed, that 0897, this is and why it's called ORF? When there is no function, then they called ORF. ORF is open reading poem. That means it is coding for something, but the function is not known. So we showed, yes, this protein helps the DNA-B to load, and therefore it can happen. Now seeing is believing. How do you know helicase is hexameric? I just told you. Now see, this is helicase. Can you see? 1, 2, 3, 4, 5, 6. So each of this unit, 1, 2, 3, 4, 5, 6. So this is a helicase protein and it's a hexameric. And what is this cavity? This is where the DNA moves after it loads. And that's what I told you, a circle inside the circle. So DNA moves through that, and seeing is believing, and this is through electron microscope we did. We found that this happens, helicase is hexameric. And we also found that when you add this protein, which I have told you, 0897, it enhances the activity of helicase. Why? Because now it helps this protein to be loaded on DNA, and therefore the helicase activity is increased. Now imagine, this is our model. So this is the helicase, and this is helicase loader, which is required to be loaded on the DNA. Now if I have an inhibitor which can inhibit the loading, then the H. pylori bacteria cannot grow, because the helicase has to be loaded on DNA, but helicase cannot be loaded on DNA without the help of helicase loader, because this is a circle, this is a circle. Now I can have a small molecule which can inhibit this process, that will inhibit the loading of helicase, and that can lead to the inhibition of replication process, and that can lead to the growth defect of the bacteria. And this is one such process we have done to show that the beta camp in Helicobacter pylori with FDA approved drug, that works in the micromolar range. Now this is also another very important thing we have to understand. These days no drug will be accepted unless its IC50 value or MIC comes in the nanomolar range. Micromolar is good for your publication, but it cannot go further unless it comes to very low micromolar or nanomolar range, and that's where the major challenge is. And that is why you know that a drug from a lab to the patient, it takes 10 to 12 years, because many times it fails. How it fails? Fantastic work in vitro. So you take bacteria, put this chemical, bacteria is not growing. Fantastic. But as soon as you put it in the body, whole scenario changes. Those who are working in the drug field, you know. Then pharmacokinetics, pharmacodynamics. What is the half-life of the drug? It's a potent drug, but as soon as you put in the body, decomposes. Not a good drug. Solubility. Major drugs fail because most of these, they are not water soluble. If it is not water soluble, then again it's a problem. How long it stabilizes, and the half-life of the drug. If a half-life is longer, good and bad, because all drugs are bad for your body. It should work and then should be excreted from the body. If it's too slow, that means it is working, but then somewhere is going, and liver is always getting it, and then toxicity will start. Or it's too short, by the time you are introducing, it is getting degraded. So in vitro, till in vitro, I am synthesizing, I am putting in vitro, absolutely fine. And that's where majorly we fail when it comes to the in vivo situation. Now imagine only in cell line. Then people, what they do? They take it to the cell line. You take cancer cell line, you take non-cancer cell line, you show the toxicity. Then it goes to the animal model. After animal model, all these preclinical tests, then it goes to the clinical. And who is doing that? Somebody has to pay for it. Who will pay for it? And that's where also major academic projects fail, because unless you have a funder, and who will fund? Otherwise company will fund. Company will never fund unless they see there is a profit. Now there is a lot of fight going on. What would be the price of the drug? Many of the life-saving drugs are very, very highly expensive. Why? Now imagine what happens. Company has put money for 10 drugs, but ultimately probably only one drug has been marketed. So what company does, pharmaceutical companies, they get all the money recovered from this drug which is in the market. But again, this drug is maybe for 2 to 3 years. After that you will see. So you see that, when you go to the doctor in the evening, a lot of people, they are basically they go and then talk to the doctor. What do they say? That the drug is slowly getting away and our company has a new drug. Now imagine now the whole system, whole chain, how it is working. So the shelf life and the half-life of a medicine, maybe 2 years, 3 years after that a new combination has to come. So you have to continuously, in most cases what happens, in academicians, you come to this level and maybe you can go to the animal testing or cell line. After that, it has to be taken by a company unless there is an institution and government institution who is interested and come here. Why I am saying this? That means it should not be that all the time we are looking for a drug. It is important to also look for the mechanism, because now if somebody has looked at it and found that this protein has the capacity to inhibit, if you can inhibit it, to inhibit the bacterial growth, then somebody else can come and also take it forward. Now after replication, what happens? Cell division, isn't it? See, there is one bacteria. First thing what will happen? The nucleoid will divide. So from one nucleoid, two nucleoid. And then the bacteria has to divide, a septum has to be formed, and then it will divide 1 to 2. And that's the normal cell cycle. And that is called cell division, when the cells divide. So when the cells divide, what happens? If you look at, we found in this bacteria, so this is from a small bacteria, and then finally you can see elongated bacteria, and they are almost divided. We found interestingly that the replication machinery in this bacteria is at the pole. Why it is interesting? Because suppose bacteria has to replicate, then it is better it replicates from the middle, because then it helps to segregate into two, and that's what a simple way of replication and division. But in this bacteria what we found, that no, the whole process is biased to the pole. As you can see here, there is a protein called single-stranded DNA binding protein, which is also a replication protein, and if you look at in this bacteria, it is at the pole. Whereas in E. coli, it is all the places. So this gave us the clue that there is something very unique going on in this bacteria. Replication is not as we see in the textbook. Any textbook you take, they will say E. coli, bacillus, and it happens in the middle. But no, it happens at the pole. Which pole? You have a flagella and non-flagella. So we thought maybe when it attaches to the cells, so there is a mechanical strength here, so maybe the application is happening here, not here. So a very simple way you can do that, you identify where is the flagella. So you know that's here is the flagella. Now you try to see where is the replication. So you try to take these two protein and try to localize where in the cell. If both the flagella and this replication protein are at one pole, that means it's flagellar pole. If it's opposite, that means it's non-flagellar. 50-50. So it's not because of the flagella which is giving to the attaching. So that means the replication can happen either the flagellar pole or the non-flagellar pole. Now, as I told you, that after replication it comes the cell division. So bacteria, it divides, the nucleoid divides, and then the cell has to divide. And there are 21 proteins to 23 proteins in E. coli, which helps this septum to be formed in the middle, and then two cells begin. However, in H. pylori, we did not see all these proteins. It is minimized. Which was more interesting, if you look at here, that normally septum forms in the middle. So the proteins which are involved in replication and cell division, they are normally in the middle. Which we found interestingly that there is a polar bias of replication as well as cell division. So you take a protein which is cell division, that is also at the pole, as you can see here. See here. You also look at the protein which is replication, that is also at the pole, as you can see here. Now you see both these proteins, they are co-localized at the pole. Nicely here, co-localized as well as here. So what is happening? In most of the bacteria, it starts in the middle and then helps to divide, and then it divides. In this bacteria, the whole machinery is loaded at the pole, and then slowly it's moving to the middle of the cell. So we looked at the mechanism and found that not only the replication and cell division protein, they are co-localized, they also interact with each other. And as you can see here, very nicely shown, replication protein, cell division protein, they initially at the pole, and obviously when the cell is dividing, they come to the middle. And this is only possible if the bacteria is moving or growing asymmetrically. And what is asymmetric? E. coli, it grows both sides, and that's why it's in the middle. But in this bacteria, it grows only one side, and that's called, many other bacteria like mycobacteria also, asymmetric growth, only one side growth. So eventually it will look like it has now doubled, but what has happened is like this. Both replication and cell division, they started at the pole. Now nucleoid is moving this way. Replication has started, so these protein move to the middle and the nucleoid move here. So what happens eventually, when half of the bacteria completely nucleoid is divided, then cell division takes place. So this is what we identified, that there is a complete mechanism of asymmetric division which takes place in the bacteria, which doesn't happen in other gram-negative bacteria. And there are protein called 0897, which I have showed you, that is unique protein which happens in this bacteria. Now it doesn't matter how long we study on bacterial replication, what mechanism we study, unless we have some molecule which can inhibit this process. So recently we have found this molecule which is called acreflavin, and this acreflavin is a mixture of proflavin and triflavin, and what it does, it inhibits the growth of H. pylori clinical isolate. Now what happens, you are using only lab isolate, but you also have to use the clinical isolate, the isolates which have been taken from the patients, and see whether it is working against them also. So we found yes, both. Now there is very interesting thing it happened, that MIC of this molecule for H. pylori, it was identified to be 2 microgram per ml, which is 10 times lower than E. coli. So you also know that gut has microflora, gut has bacteria. Why it is not suggested to give you antibiotic very frequently? Antibiotic kills the bad bacteria, antibiotic also kill your bacterial microflora. So what happens when it is gone, again it has to come back. Now if you do it frequently, and these days they say everything from your behavior to everything is actually guided by your microflora. So it's not good to use antibiotic and to get rid of your microbiota. So therefore if you have a molecule, hypothetically, which is killing the bad bacteria at a very low concentration, but not touching your good bacteria, that is better. And this is our hypothesis. Since it is working at least 1/10 of the concentration which is required to kill E. coli, so we found not only that, and also clarithromycin, another antibiotic is used for H. pylori infection. It also acts against clarithromycin resistant strain. But again, as I have told you, it has no meaning when I only take the culture and do. So can I take it and do it at least in animal model to show that yes, it works in animal model? Yes, we did and showed that it eliminates H. pylori from H. pylori infected gastric tissues. So this is uninfected, there is no infection. This is infected, infection is there. Basically you are looking at the gene of that bacteria, whether it is present in the stomach by PCR. Now if you do the infection in the presence of this molecule, the infection is gone. And you can see this way, now this is the biopsy, and then do the immunohistochemistry to see whether it is protecting. Yes, this is control, no infection. Now see what happens to your gastric cells. It pinches, it goes down. Now when you again treat with this, now this is as good as this one. Not as good, but at least it is similar to the control. So that tells you this molecule, it not only works in bacterial culture, it also works in vivo in a model for H. pylori infection. Now what is happening, how it is happening? We found that this molecule, as you can see here, it actually completely disrupts the membrane of the bacteria. As you can see here, this is EM to show that this, in the presence of this molecule, you can see this. So the membrane is getting disintegrated. Now of course this mechanism we need to find out. Why at this concentration? That means there could be some specific target in the H. pylori which is not present in the E. coli, which is getting targeted by this molecule, and at a low concentration also it can work. So this is what we have found. SCF-HCl inhibit H. pylori growth, and it gets synergistic effect with antibiotic clarithromycin, and this treatment completely eradicated H. pylori infection for in vivo mouse disease model. And just last week, 2 weeks back, we have got an Indian patent. Took us almost 6 years to get this patent. The reason being SCF-HCl was shown earlier, because if somehow those who have patented, you know, if there is a prior knowledge, then you won't be able to patent it. But what we showed, that this molecule can act against a clarithromycin resistant bacteria, H. pylori. So that was not shown before. And we also showed, it showed synergistic effect. Synergistic effect is what? One drug is causing this much down, another drug is causing this much down. When you mix this, it is causing this much. So it is not 2 plus 2, 4, it is actually 6. So somehow this synergistic effect means when they combine, they show much higher effect. So that also we found. And now is the real challenge. Because I have done what I could have done in my small lab. Now it has to go to the clinic. Now thorough preclinical studies have to be done. How toxic or how bad this is for our body. And then it has to go to the clinical trial. So someday, if I can, maybe I still have 6-7 years, I will see if I can take it to the next level. It took me almost 25 years to come to this stage. Now another 5-6 years, I will see if I can take it to the. Now I will end with one more minute. Along with this, we are also trying if we can have some natural compounds from plants which can also take care of H. pylori infection. And in Northeast, particularly in Manipur, those who are there, and they have healers, traditional healers. What these healers do? They are not doctors, but they have the knowledge of the medicinal plants, and they give you these plant extracts, or sometimes they will give you the root, or they will give you the bulk, and then they will treat you. So there is a tremendous knowledge from these traditional healers which has not been tested in the laboratory. So we collaborated with IBSD, Institute of Biodiversity and Sustainable Development, which is a DBT institute in Manipur. Some of you, those who are from Northeast, maybe heard of this institute. And they gave us 20 such plant extracts which these traditional healers use for gastric patients. Those who have gastric related problem, they use that. So we took those, and then one of them is xanthoxylum, which showed a good start of low 2 mg per ml MIC concentration both in liquid culture as well as it is in plate culture. Now you can say, again, interestingly, that extract does not affect E. coli. So again, it's a good control that this extract has something which is killing the H. pylori, but it is not killing the E. coli at that concentration. Of course, if you increase the dose, everything will be killed. And also, not that much of toxicity of mammalian cells which we have found. Now you will trash it, anybody will trash it, because it's just an ethanolic extract. There will be 100 molecules there, and anything can do that. So what you do? Now this is what we have found. Some of them are really encouraging results we have got, and then we have now done the LCMS. So through mass spectrometry, you can now separate those molecules, and now we have got around 20 molecules which we want to take it further, whether any of them or in combination can show the anti-H. pylori activity. And now you can think, out of these 20, how many combinations will be there? But there are some good scores for some of this. I will start with that. First you have to buy those chemicals, then test it in vitro, then in vivo, and then in combination, but we will do. The reason being, you should never stop. If you get something, take it forward. I will stop. It is exactly 11 o'clock, 5 minutes have exceeded. So this is my group in SCMM. Now it has reduced because these days only thing I do is administration. I have joined this institute as a vice chancellor. So my target is getting student and admission, not H. pylori. But I do come to the lab every evening. I spend 1 to 2 hours every day. Saturday I spend. And all my students, past and present, all my collaborators, as well as the funding agency. And I'm sorry if I have bored you last 1.5 hours, but I tried to give you my journey, which we started in 2000 in the lab, and we have made little progress, and hopefully by the time I retire, I can say I have 1 or 2 molecules. If yes, I'll be really happy, because that's no awards actually can compensate that. Awards are good for you to progress, but eventually it is that consolation and that satisfaction. If I can do something, then only it will be fulfilled. Thank you so much. If you have any question, please feel free. Yourself. If you have continuously this problem, please visit a doctor, because we are fake doctors, we are not doctors. You have to see a real doctor and get it checked. It is important. Acid reflux can happen for many things. But whether it, imagine you are continuously acid reflux, and if you have already H. pylori infection, so what happens, as I told you, that 50% of the world population harboring, but disease may happen only 2 to 3% below that. That means it is your genetic makeup. It is also other factors. But should you wait that long? No. If you have a problem, you should go and then see a doctor. And if you already have started getting those symptoms, then definitely you should go. Yeah.
Sir, I have 2 questions. One is related with the climate change in India. As you told that the climatic parameters changing, the temperature, moisture. So what the biological things changing that make any bacteria is the multi-drug resistance? And second question is, what are the challenges we are facing for the vaccine in the case of
helicobacter? Very good questions. See what happens in what are the ways a strain or a bacteria or a pathogen become resistant? One, drug goes inside, it metabolized. The bacteria has evolved a pathway to metabolize the drug. Second, it pumps out the drug. What happens? Drug goes inside, but then it pumps it out. If it pumps it out, so there are transporters are also there, and this is very common. What happens in case of resistant? Now the drugs are throwing out of the. So now these transporters were there, but with changing time, because what is happening, as you have to imagine, with climate change, the physiology of the bacteria is also changing. And of course, the bacteria also needs to survive. In case of SARS also we have seen. So initially what happens, they are more virulent. Then the virulence goes down. And the duration also, earlier maybe it was, as they said, 21 days. Now it's only 7 days, but the peak comes. So they are also trying to go as many places they can go, reinfection, so that they can survive. So it is because of, or what happens, there is a particular gene or protein which was getting affected by this antibiotic. It could be translation, it could be transcription, it could be replication. But now the bacteria make a mutation. Now if the protein is now mutated, now this antibiotic won't work. And now that is what is happening. It is a Lancet study very recently. They have shown that in last 10 to 15 years, 10 to 20% of the AMR may be contributed because of the climate change, because of the changing behavior of the microorganism, adaptation in a new place, change in physiology, all will contribute to this. Now if you say temperature, how it is only one bacteria, I cannot say. What they see, what was the temperature or climate or other parameters 10 years back, what is now in this area, and how many bacteria and how much resistance has come. Now, excellent questions. Why there is no vaccine? Now what happens, there is an argument in this field. If bacteria is there in 50%, 60% of the population, do you want to take them away? Is it doing something good also? Because if it was found only in people when there is infection, not others, then it makes sense. You have to understand now what are the mechanisms by which a bacteria which was not pathogenic, now after 3 years it becomes pathogenic. Because I can guarantee, if I do test all of you, at least in Indian context, because of the socio-economic condition we live, half of your people will have this bacteria. But possibly some of you are at the borderline in 5 years or 10 years, and it also complicated by your food habit, by your overall lifestyle, because everything these days are lifestyle. How good or bad food you are taking, and your genetic makeup. Maybe you are strong enough, your immune system is strong enough, your genetic makeup is strong enough to protect you. Those who are not, they will fall prey of it. But my suggestion would be, I myself have gone through twice. When I was a student here, I was infected with H. pylori twice, and it's a hell when you get that. So thankfully now I am treated, lot of antibiotics, but if you have the symptom, it's very common, please go and see a doctor and get it treated. Yes?
Sir, what is the natural reserve of this bacteria? Before it comes to the human, there must be some. How does this evolution work, like to adapt to the US condition?
Very good question. Possibly this is the only bacteria for which maximum number of strains have been sequenced. You take any animal, it is there. Cat, dog, anything you name it. So therefore, it is there forever. But so far there is no incidence that a strain from dog or cat can be colonized in human. So therefore the evolution of the human strains possibly has happened in such a way they can adapt in the human body. But it is there in all animals. Whichever you take, you will find that.
Outside any animal host or
That's what I'm saying. No, it is not through, it is fecal oral. Through contaminated water, through contaminated food, it can come. But it is not through. So it has been shown that, so what they do basically, suppose I take a strain from a cat and put it in mouse and see whether colonization is happening. Because this bacteria, first it has to colonize. And what is colonization? Any foreign, anything bacteria or any microorganism comes, first it has to replicate and get to a concentration which now superpowers the immune system. Isn't it? Otherwise immune system, see, imagine if through a mosquito, when a mosquito bites, suppose a parasite, it injects only 2 to 3 microliter. And what is the volume of your blood? 5 liter. So it is diluted, it will be taken care of. But it multiplies. So it is not, so far has been found that any other animal, but in human, from 1 to 1, it is basically through, and contact also. Even like this is the mostly studied genomic sequence bacteria because of its prevalence and its importance, particularly in cancer. But there is no incidence, at least I don't know whether cross infection can happen. You had a question? Yes.
So, sir, I just want to know that as you said that it may be present in many of us, but it is only infecting causing these symptoms in some. Sir, apart from ulcer and cancer, does it have any other impact or some other conditions which are related to
Yes. So these are all correlation, direct correlation because of its niche. It is with gastric, but it has been now shown the connection with other diseases. But how it is happening, those have to be. So this is correlation. Suppose I get some like coronary patients and look at their profile. So there is a strong correlation of many of the diseases with this bacteria. But how it is happening, those mechanisms have to be. Because imagine if 50% of the people are having, not 50% having stomach diseases, they might have other diseases. Now whether this is also causing. So that is possible and there are correlations, but right now I cannot tell you any definitive that this is for another disease.
Sir, one more addition to this. When somebody is having ulcer, maybe, so if the condition persists, if it is not cancerous, but still not cancer, but ulcer, so can it be like, it may have some other related condition, maybe autoimmune or something?
Sorry?
Autoimmune can be like some condition.
See, ulcer if you have, it's treatable. And it won't go unnoticed. You will have pain and you have next thing you will have blood in the stool and it is treatable. Now, the body, that's why people say that it's all systems biology. One problem in one place is not that it will stop there. It can lead to other problems, but how and when, that depends. Unless you study it thoroughly, it's very difficult to say. But very much possible it can lead to other complications, and particularly if it's untreated. So therefore, if you have symptom, please go see a doctor and get it treated. Yeah.
So one thing comment I want to say that I also suffered once from H. pylori infection, and the doctor at that time told me to blow the balloon and get it. So today I understand
How it happened. So that is exactly my job was, that what happens in the lab, how it happens. So now you know why you were asked to inflate that balloon.
Now I'm clear this time. My question is that is there any for COVID there are different variants, isn't it? So only one type of H. pylori?
No, many strains.
So for there we need to
Yes. Even in India, from northern part, southern part, there's lot of strain variants. And also, disease causing ability is also different. So there are not, North India there are studies, they have looked at these strain variations. Now we are trying to connect with Kashmir. So Kashmir, they have got a big grant to look at, because in Kashmir the occurrence of gastric related disease is much higher than rest of the India. So now they are looking at, so they connected with their food habit and different type of, they take this salt tea also, all these things, but now they are trying to sequence these strains and see how. So this is what you are saying is basically epidemiological study. We are looking at very basic, but that's also very important when it comes to treatment, like which region you will treat by which regimen of antibiotic and other. So there are lots of, those who are from biology and PubMed, you just go and type H. pylori, you will see how many papers and how many strains are there. So there are lots of different strains, and not only that, geographically also they are different. Asian strains, then North American, then also European, there are lots of differences are there.
Variation from which, it looks similar in imaging. So is there any, how do you define which is, there is different variant?
Okay, so I told you about that CAG-A, VAC-A. So these are the pathogenic factors. So they look at the variance in these pathogenic factors, because variance in those pathogenic factors will lead to variance in infection and outcome. So people look at those genes, and also standard 16S RNA, people look at those.
On this particular H.097, it is only found in pylori.
H. pylori, yes.
So is it reported, working on this?
Yeah, we are trying to make small molecule inhibitors in 0897, and if you can block the helicase loader, that's what we are trying, but it's not a very good expression we get, so we are still trying. And it's not an enzyme, so enzyme are easier to inhibit because it has active sites and you can block, but it's not an enzyme. Only thing you can do, the surface which is interacting with the helicase, if you can block that somehow, so that would be a good inhibitor. So that's what we are trying. Okay. Sorry, ma'am.
Conventional technique to detect infection by the
Okay, so there are dipsticks these days. So, but this is based on serum. So they have to take the serum, and then this is through the capillary. They do. I showed in my slide also. And but otherwise, as he mentioned, no, this is like they will give a bacteria a tablet which is C13 level urea, and then you will inflate. So that is non-invasive. And if this is C13 CO2, if you are doing, they will just count that and they will tell you. And there are these diagnostic kits are also available for that, based on urease. So very safe, because no other bacteria has this enzyme, so the detection is very robust. Oh, that is based on that exactly, and also that scintillation counting. No, they take that and then first they enhance. So there is a technique there. I don't know exactly that physics of that. But any other like isotope as you can detect, you can do that. Okay? Okay, thank you so much for your attention. Thank you.